d'accueillir Helen DONOGHUE, qui est une collègue de Grande-Bretagne et qui est une des personnes qui a lancé avec nous ici le champ finalement de la paléomicrobiologie. Comme elle va vous le montrer, elle a travaillé plus particulièrement dans le domaine des mycobacterioses anciennes, la tuberculose, la lèpre dont elle va parler aujourd'hui. Et donc j'ai beaucoup de plaisir à l'accueillir à nouveau, puisqu'elle était venue il y a quelques années déjà nous donner une présentation plus sur la tuberculose. Et donc it's a pleasure and honor also for us to welcome you again in Marseille, so that you can give us some insight into the palaeomicrobiology of leprae. Thanks again for coming today.
Well, thank you for the invitation, and I hope you understand my English. My French goes back so long I've forgotten almost all of it, so thank you for your tolerance. I was actually on the very first paper that demonstrated it was possible to examine archaeological material and to find evidence of M. leprae ancient DNA, and that was published in 1994. So I've been doing this for quite a long time. Now, just some basic facts about leprosy. It's a gram-positive rod, and in the States they call it Hansen's disease after the Norwegian leprologist who did much early work. And one of the things I'm going to be talking about are the cell walls, because they are very characteristic as well as the DNA. But these cell walls are so thick and resistant to the environment that it is believed that they may contribute to the exceedingly slow growth rate of the modern day organisms. Certainly, M. leprae are wonderfully preserved in archaeological material, and I find them better than M. tuberculosis. Even now, there are about 200,000 new cases of leprosy a year, and leprosy is the greatest cause of disability arising from a bacterial infection. So it's an obligate pathogen, and although I say here it's an obligate parasite of humans, in the laboratory they can be passaged in other organisms. The experimental animal is the 9-banded armadillo, and that's used to produce reagents, but it can be passaged in mice foot pads and even in amoebae. It is a nerve disease, and the organisms bind to the nasal epithelial cells and to Schwann cells. And you get very characteristic changes, especially to the hands and feet, and that enables us to recognize the disease from the skeletal remains. And I've been doing work mainly in Eastern Europe, and I'm also currently looking at a large number of samples from North Africa in the Dakhla Oasis, which is about 200 to 400 AD. And at the moment I'm getting about 20% of these cases positive for leprosy. They were sent to me by El Monte, who some of you will know. And the modern disease, it's a disease of extremes. The clinical manifestations depend on the host immune response. If you have a good immune response, you get paucibacillary or tuberculoid leprosy. If you have a poor cell-mediated response, you get a humoral response. Very high number of antibodies produced, but they're useless. And you get unfettered, unlimited growth of the leprae bacilli. And you can see you get a different cytokine pattern depending upon which which part of the spectrum the individual patient is in. And the problem with leprosy is it can be treated with anti-mycobacterial drugs, but if you wait too long, you get an autoimmune reaction, and it is the autoimmune reaction that can lead to the damage to the human nerve tissue and permanent disability. These are some photographs of patients. I actually went with a Spanish group to Honduras in Central America, and these were some patients who had unfortunately been abandoned for 20 years due to civil unrest, and they had only had monotherapy with dapsone. And you get cataracts, you get resorption of fingers and toes, loss of the thumb, and in the past, there's plenty of historical evidence that people were ostracised and excluded from society if they showed these symptoms. Even now, it happens. Right, if we get to the bones, again, there are particular sites that we always look at. Around the nose and the maxilla, and then the extremities, the hands and the feet. And one reason for this is that the optimal growth temperature for M. leprae is around 30 to 32 degrees, and that may be why you find it at the extremities where it will be a bit cooler. In archaeological material, there's no treatment, and so if somebody loses all sensation in their hands or feet, you will always get secondary infections due to damage, burns, cuts that aren't detected and get subsequent infection. So we have to be very careful. We're looking at leprosy and not secondary infections. Right. This is what leprosy looks like in a skeleton. And here we have leprosy from a Byzantine site inland from the Black Sea from about 800 AD, 8th to 9th century, and you have resorption of the nasal region, and you get generally resorption of the maxilla, the top teeth drop out, and eventually the palate gets resorbed, and so the mouth and the nasal cavity become one large cavity. This is the oldest skeletal evidence suggestive of leprosy, and it's from India, 2000 BC. Lots of us have looked at specimens from this skeleton, but unfortunately it was stored under very hot and wet conditions, and if there was any DNA there certainly isn't now. But it does show the typical signs of the loss of the resorption of the maxilla and the rounding and enlargement of the nasal cavity. Now, I do a very, very long, slow extraction, which you needn't take detailed account of. The main thing is that I don't do an extraction in less than a week, and very often it goes on for about 10 days. And one reason is because you're trying to break up this very thick, tough bacterial cell wall. We also treat it with a particular chemical, which is this substance here, PTB we call it. It was first diagnosed in the plaques of the brains of people with Alzheimer's disease. And it stops covalent bonds, and it breaks covalent bonds. So it kills any PCR, but in the extraction, it can let you release DNA that may have got cross-linked over time, and it definitely helps if you put it in. And if you're detecting ancient leprosy. It helps if you've got a multi-copy site to target. So if there's some damage, there might be a corresponding sequence elsewhere in the material, elsewhere on the DNA from the organism. And so we normally screen with this multi-copy target rLep, where there's 36 copies per cell. That's to help us have a chance of finding it. And there's something called replep, where there's 15 copies per cell. And these are quite good for looking for M. leprae. One thing we discovered was many people with ancient leprosy are co-infected with tuberculosis. There was a bit of skepticism when we first showed this, but we've now got very nice evidence. It makes sense really, because if somebody's got leprosy doesn't kill you very quickly, but it makes you very sick and unwell, and if you're somewhere where there's a lot of endemic TB, then that's what kills you. And we routinely screen for that as well with repetitive sequences. So insertion sequence 6110, there's between 1 to 25 copies per cell, and IF1081, there are 6 copies per cell. And so we look for those first in a screening test, and then if we get any strong results, then we start looking at single copy sites and try and do genotyping, etc. Now the thing about ancient DNA is that it fragments over time. And what the histogram on the bottom left shows is that the smaller the target sequence, like 92 base pairs, compared with that, anything larger, you get a smaller recovery. So the smaller the target sequence, the better result you get. And with conventional PCR that we were doing until about 3 or 4 years ago, you detect your amplicon by running a gel, and we had to have special agarose that enabled us to actually pick up bands of 92 base pairs, because normally you don't get bands that small. Now we use real-time PCR, and we use the QIAGEN rotogene, and here we've got specific primers and probes. So the probe has fluorescence that is blocked, and if it binds to the amplicon, then the fluorescence is released, and so you can follow the reaction in real time. So here we have real-time PCR, we have dual-labeled probes, and that lets us get down to about 62 base pairs. So it's very good for ancient DNA because it's so fragmented. And these are some samples that I looked at from Hungary, dating from the 7th to the 10th centuries. And basically, I'm sure you know this, the earlier the cycle threshold, where it starts going logarithmic, the more DNA you've got there. And looking at the same samples, we get comparable results with replep. This one didn't have any cross-infection with TB. Now, M. leprae has got a very reduced genome. Its genome is about 43% of that of MTB. And it's got very poor DNA repair. And there are many repetitive units in M. leprae, and we believe that it is possible that even during the course of a long-term human infection, you can get changes in the number of repetitive units. There have been papers published demonstrating in one patient different numbers of repetitive units in the nasal region compared with the hands and the feet. Now, it could mean you've got a mixed infection at the start, or it could mean that you've got some slight selection going on, perhaps because of the different body temperatures at the different sites. But anyway, the ancient DNA from some Hungarian samples was so good that we were able to actually distinguish between different leprosy cases by looking at repetitive units and seeing that in one case we've got, say, 12 repeats of the sequence TTC, and just across the cemetery, slightly later, we've got 17 repeats. Now you imagine you need very, very good DNA preservation to be able to pick this up in something that's over 1,000 years old. But with leprae you can. Now, the strategy that I've adopted over time is to have more than one method of looking at archaeological material, because when we first started, it was in the very early days of PCR, and there was a huge amount of skepticism. So as soon as I started saying I was finding M. leprae, or initially M. tuberculosis, in bones with no lesions, paleopathologists who are trained to look for lesions, they're not microbiologists, they didn't understand that you only get bony lesions in about 5% of every TB case. It's a minority thing that you get. You have to live long enough, you have to have disseminated TB. There's not that many people in that situation. So the majority of people with active TB will have no signs on the bones at all. That went down like a ton of lead. I don't know if that's an English idiom or whether you have it as well. When I first started saying that we had TB from cases with no bony changes. So what we started doing, originally they said have somebody else do the same work in a different lab, but we decided we would have a totally different method. We'd have an organic chemical method, and so I linked up with a group at the University of Birmingham, Del Bezra's group, who are doing lipid analysis using traditional organic chemical methods. Originally they were doing gas chromatography, then mass spectroscopy, now it's MALDI-TOF, all sorts of these high throughput techniques that are very sensitive. And so we do it in parallel, and we just confirm each other's results. And the lipid markers are much better for the older stuff, but they don't give you quite as much information. And the main problem is they can't give you genetic information. So originally we had the morphological analysis. Some people have done histology, not that many people. Proteomics, we've just submitted a paper looking at proteomics of some 18th century Hungarian mummies that I've been working on for a large number of years that have got a lot of DNA in them. But the proteomics, well, it's very iffy. So I think we can say we're not really up to speed on analysing proteins as direct evidence of bacterial infections in the past at the moment. We've got some initial results, but they aren't that thrilling. So we've been going for PCR, next generation sequencing and whole genome sequencing. I don't do that, but luckily, because I've got the samples, I've got lots of people wanting to collaborate with me, and they've got the methods and the money to do it, which is great. Now this is what it looks like if you're looking at cell wall lipids. So if you look here, we're looking at mycolic acids. These are in mycobacterial cell walls. And these are different. This is body 18 right and left. ribs, and body 144. These actually are published now, and they're from northern Italy. And here we have MTB and leprae. And you can distinguish these by the cell wall lipids, and in fact what we've got is M. leprae here. But you can pick up mixed infections. So here, if you separate the mycolates, you do alpha, methoxy and keto mycolates, you don't get methoxy mycolates in M. leprae. But you can see, I hope, that if you've got standard M. leprae, MTB and our strain, there are particular peaks that you get in M. leprae but not in MTB. So you can tell by looking at the cell wall lipids whether you've got tuberculosis, leprosy or a mixture of both. And this particular one, which was from I think 7th century Hungary, we've got both. There are more complicated and more hydrophobic lipids, and the mycocerosates are the next group that David Minikin and his group started working on. And here again, we have, if you look at the peaks, his postdoc came up with this. So we have an L for MTB and a crescent for M. leprae, and here we've got both, showing we've got a mixture of both bacteria. And they are finding more and more of these very long, hydrophobic, complicated cell wall lipids, and more of them are now getting identified and characterized to be used as further lipid biomarkers. Right, now I'm interested in ancient TB and leprosy because it tells us about human migrations. This was from a paper, it was a commentary in Nature Genetics about a paper by Mono et al. Looking at genotyping from M. leprae from around the world, and basically showing that if you've got a very mixed population and you then have an evolutionary bottleneck, you can end up with different clones of pathogens associated with different human populations. And that's what we find in both leprosy and tuberculosis. So I'll give you some examples of that. But before that, the basic way of genotyping M. leprae, it only got worked out after the genome was obtained, which was in the early 2000s. So a paper came out, again by Mono et al. In 2005, where they basically worked out 4 principal genotypes based on three loci here, and they weren't quite sure which came first or second, but you've got here examples of differences between different strains from different parts of the world. If we look at European leprosy, almost all of it is genotype 3, but there are a few cases of genotype 2 which have been found in Denmark, Sweden and southeast England. But otherwise, they're really all genotype 3. But genotype 3, there are many different types of it. This was the map they originally produced in that very first paper, and it's suggesting that there may have been an origin of M. leprae, perhaps in the Indian subcontinent, but then they weren't sure whether or not that might have been the origin. You know, they weren't sure which came first, But what they were sure was that West Africa slave trade led to leprosy getting into the Caribbean, and the European leprosy getting into North and South America. But there's some doubt. We think it may have started in the Horn of Africa, and of course that's where we think humans may have originated. This is the follow-up paper in which I and my colleagues were involved. And so if you've got very good eyesight, you may see that some of these little pillars have little stars, little asterisks at the top. And these are all based on ancient DNA data, because European leprosy is extinct, but we have got evidence of it from the archaeological material. And this time, the authors were subdividing the genotypes and even trying to put together migration patterns around the world, like that is the Silk Road, and they weren't sure whether it came from east to west or west to east and then back again. There's been lots of interesting suggestions about how it's got around the world. But what we do know is that in the New World, it basically came from Europe or West Africa. I dug this out of a Google search looking at movement of peoples, because the Hungarian cemeteries and the early material I've been looking at were associated with this group of migrating people called Avars. Avars are thought to have come from somewhere near Mongolia. They were a bit like the Huns or the Magyars. They travelled on horseback. They were moving westwards, looking for new places to settle, causing a lot of upset, you know, killing, raping, etc. But eventually they settled down, and there was a huge Avar Carginate, that's what they called it, where I've been looking in Hungary, Austria, Croatia, that area. They actually besieged Byzantium and wouldn't move until they got a huge payment in gold. And one of the individuals I looked at had been buried with his horse, and he had one of these gold pieces. So we have a nice direct link with migration and particular people. So we know they came via Byzantium. And we have our Turkish sample I showed you earlier was from the Byzantine period. So it all fits quite nicely. Right, my most recent paper about this was published earlier this year, and it includes a lot of work done by my collaborators as well. And so this area here is the area that was still part of the Avar sphere of influence. So the red dots are 10th to 11th century, the green dots are earlier, and this is 700 AD, this is where the Avars were, but we've got some Avars in Italy as well, and there's our Turkish Byzantine one. And there is the earliest known case of leprosy, which is from Uzbekistan. That one is yet another genotype. It's not the one that I've been finding in Hungary. Right, and just to show you what it looks like if you've got both diseases in a skeleton, this is an 11th century Hungarian, where we've got the rounding of the nose, loss of the palate, loss of the maxillary teeth. The palate is getting eroded away and will eventually disappear totally. And we've got these lesions in the spine. And we were able to show that this individual had both tuberculosis and leprosy. So we got the paleopathology for it as well, and the cell wall lipids. Right, this is some work which is newer, but I've talked about it in Bordeaux. There was a biomedical sciences conference there. And this is work I've done with Joel Blondeau, who is based in Normandy. Yeah. And there's a leprosarium where you've got several human remains buried in very simple homemade coffins, but you've got very definite signs of leprosy, like collapse of the bridge of the nose as well. And here's another example. So this time it's from Normandy. Being in France, I thought we'd like to have some French examples. So we've got the rounding of the nose, we've got the total collapse of the central part of the maxilla. That's what you get in very late leprosy. And then we've got all these spinal lesions. And this is, as you see, a young woman, and we've got DNA and lipids from these. Right. Now, I've been doing a lot of genotyping with these because they're very well preserved. And to do genotyping, we just have a general amplification using cyber green as a basic fluorescence, and then a bit of temperature analysis, and then we have the sequence. And we're looking for single nucleotide polymorphisms. So here we are, so this is one of the Norman ones, and we have C, G, C, the particular loci, and that tells us it's genotype 3. So that's what we were doing last year that I talked about in Bordeaux. So what we've done so far over the last 15, 20 years is that we've looked at quite a lot of leprosy from southern and central Europe. And there's a definite link with the Avars. We've got the earliest known leprosy cases in southern Hungary and Austria. And we actually showed that out of 26 individuals with leprosy, we found that 6 of them had TB as well. And the summary of what we found from Normandy is that we only found M. leprae DNA in the most recent material from about 1400 AD. And we think the reason why the earlier material that looked as though anatomically they should have had leprosy there, we think it was just the environmental conditions that didn't allow the DNA to persist. And so far, at the time I did this slide, we managed to show that 9 individuals had M. leprae genotype 3. That's the basic one without the subtyping. So, to give you an idea, we're not really sure whether it's so pretty as this. This is what the 2009 paper looked like. And it looks as though it goes from A round to R in a nice sequential way. We don't know whether that is correct or not. But for the 2 Fs, there's published papers from, there we are, from Denmark, Sweden, and Winchester in the United Kingdom. That's southern England. That's the only genotype 2 that's been found in Europe. So it's tempting to think that perhaps they came from a different route, perhaps via the northern route, perhaps from Siberia or something. We don't know. But then we've got 3I from the UK, Sweden, Denmark, and France, which isn't published yet. 3K, 7th to the 10th centuries in Hungary and Turkey. We've got this Uzbekistan 3L. We don't know what the Egyptian one is yet because it's so fragmented, we haven't been able to genotype it completely yet. And then we've got genotype N, which we found in the Czech Republic and 10th century Hungary. So it does give you an idea that we're getting different subgenotypes in different parts of Europe. Right, I think this is where I want you to stop filming.